A warm welcome again. So we are in third session of our video lectures. Uh, in the earlier sessions, in the first session or the session number 1, we discussed about classification of microorganisms, difference between a prokaryote and an eukaryote, structural components of a prokaryotic organism and structural components of eukaryotic organism in the session 1. And in the session 2, we discussed about mitochondria, Golgi complex and endoplasmic reticulum. Three major intracellular organelles which are all double membrane bound organelles present in the eukaryotic cell. So coming to the current session, we are going to discuss about another set of three major intracellular organelles present in the eukaryotic cell. First one is called lysosome, second one ribosomes, and the third one called cell membrane. Cell membrane is also a intracellular organelle. We can call it as a intracellular organelle. So moving to the first one, that is lysosomes. So among all the intracellular organelles we discuss, this one is the unimembrane bound structure or single membrane bound structure or a type of intracellular organelle. So this lysosome is also called as a suicidal bag. Why? Because they helps in destructing either the entire cell or restricting a specific type of intracellular organelle or destructing a foreign pathogen that is coming or invading the cell. So that's why this is called as suicidal bags or sacs. So why these are called as bags or sacs? Because they are round in shape and they are unimembrane bound structures and they have so many varieties of enzymes present inside the structure, bag or sac, right? So what are the different types of enzymes, lysosomal enzymes that are there? First one is called lipases. Lipases are the enzymes which has the ability to break or chop down lipids. Large lipids can be digested by lipases, and amylases are the enzymes which can break down complex carbohydrates into simple sugars, and proteases. Proteases are the enzymes which can break down large proteins into individual amino acids or small peptides. Nucleases are the enzymes which has the ability to attack the nucleic acids, DNA and RNA, and break down the genetic material. And the phosphoric acid monoesterases. These are also very important in ATP synthesis as well as ADP formation. Right? So these are various types of lysosomal enzymes which are present in this sac. Okay, and other major important function of these liposomal enzymes is these are called as acid hydrolases. That means they can function, these enzymes can work only under low pH or acidic environment. So inside the bags or lysosomal sacs, the pH will be low, that is 5 or below 5. Only in such conditions these enzymes can work. If the pH is above 5, if the pH is 7 or 6 or 8 or 9, these enzymes cannot work. So that's one major advantage, because so if there is any accidental leakage of enzymes from the lysosome into the cytoplasm, then also this cannot randomly destroy the cell, because inside the cytoplasm you have a pH range of 7 to 7.4 or 7.5. These enzymes cannot work in that pH. These enzymes require acidic environment or low pH levels for these to work. So only at pH 5 or less than pH 5 these can work. That's one major advantage of these acid hydrolases. And coming to the functions, major functions of lysosome, first major function is intracellular digestion. That means inside the cell, if the large carbohydrates or proteins or lipids or fat molecules need to be digested, then the cell uses this particular organelle, lysosome, to break down complex carbohydrates and all the other biomolecules into simple molecules so that they will be used by other organelles like mitochondria for synthesizing the cell energy currency, that is ATP. Okay, so the digestion of large molecules, large biomolecules, carbohydrates, lipids, and proteins is done with the help of lysosomal enzyme. First major function. Second major function is autophagy. Autophagy means if a cell wants to kill itself, or if a cell wants to kill a particular intracellular organelle, then the cell can employ this particular organelle called lysosome to go and attack that intracellular organelle and nullify or kill it. So that is autophagy, or the cell wants to commit suicide. Then again, the cell will release large number of these lysosomal enzymes, engulf entire cell and commit cell suicide. That's also one kind of cell death mechanisms. And third one is waste disposal, right? So waste disposal as a recycling center. Whatever the toxins that are synthesized inside the cell which need to be broken down, that will be disposed into these structures, lysosomal sacs, and the waste will be digested and converted into harmless substance. Then it will be disposed out of the cell from the cell membrane. Okay, so waste disposal is also done by these structures called lysosomes. Nullification of the toxins and make them harmless and disposal is done by the lysosomes. Next one is defense against the pathogens. So if at all any microorganism like a bacteria or the virus is invading a particular cell, right? So when the bacteria has invaded a cell, what the cell will do is the cell will use these lysosomes to engulf this entire bacteria. The bacteria will be engulfed inside these, or the virus will be engulfed inside these lysosomal structures and completely destroy the bacteria, like how macrophages work, right? Similarly, glycogen breakdown of large glycogen or carbohydrate molecules into simple sugars is very much essential for producing the energy. The energy production will happen only when glucose is present inside the cell, right? So glucose will undergo glycolysis and the end product will move on to the TCA cycle and to electron transport chain. Finally, ATP is synthesized, right? Even though NADPH and H plus or FADH also produce, all these currency molecules again goes to the mitochondrial matrix, and again they will be converted into the ATP, adenosine triphosphate. So these are the major functions of lysosomal enzymes. So let's move on to the next organelle, that is ribosomes. So ribosome is an organelle which is also called as protein synthesizing factories, which are present on top of the endoplasmic reticulum. So these ribosomal structures are studded on top of the endoplasmic reticulum and helps in synthesizing the protein. So I have discussed in the earlier class, in the earlier session, that ribosome protein synthesis require a specific messenger RNA coming from the nucleus. Once the messenger RNA binds on to these ribosomes, and the protein synthesis happens on top of the endoplasmic reticulum. So let's have a look at the structure of the ribosome. So what are these ribosomes made up of? Ribosomes are made up of only two things, that is rRNA, also called as ribosomal RNA, and proteins, also called as ribosomal proteins. Only two components are there in the ribosomal structure. Ribosomes are made up of rRNA and proteins. Okay, and There is a clear distinction between 70S ribosome and 80S ribosome. We have seen in the earlier class, prokaryotes have 70S type of ribosomes and eukaryotes have 80S type of ribosomes. Any ribosome, either it's a prokaryotic or eukaryotic, is made up of two subunits, a large subunit on top and a small subunit. A large subunit and a small subunit. In case of prokaryotic ribosome, it is made up of 50S subunit and 30S subunit. 50S as large subunit and 30S as small subunit. S stands for Swedberg unit. Swedberg unit is the centrifugation unit that gives the numbering for the subunit. Okay, 50S subunit and this is 30S subunit. Whereas in case of eukaryotic ribosomes, we have 60S as larger subunit and 40S as smaller subunit. Okay, so let's have a look at the clear distinctions between a 70S and an 80S ribosome. Organisms, which organisms 70S ribosomes are present? Mostly in all the prokaryotic organisms, we have the 70S ribosomes. Only major intracellular organelle present inside the prokaryotic cell is the ribosomes, and whereas in case of eukaryotes, we have 80S types of ribosomes. Subunits, what type of subunits in 70S? 70S is made up of 50 and 30 as we've seen here, and whereas 80S is made up of 60 and 40. Okay, composition. What is the composition of prokaryotes and eukaryotes? In case of prokaryotes, 70S type, we have more RNA and less proteins. We have 60% of total RNA and 40% is made up of protein. As I said before, the RNA ribosomes are made up of only two components, ribosomal RNA and ribosomal proteins. 60% of the 7 prokaryotic ribosome is made up of ribosomal RNA and 40% of the prokaryotic ribosome is constituted with the proteins, ribosomal proteins. When it comes to composition of the eukaryotic ribosome has less amount of ribosomal RNA and more amount of ribosomal protein, 40% of ribosomal RNA and 60% of the proteins. Coming to large subunit, in case of prokaryotes, large subunit is again made up of two types of RNAs, 23S and 5S, which we can see here in the picture as well. And a large subunit of eukaryote is made up of 5S, 5.8S and 28S. Okay, these are three different types of rRNAs, ribosomal RNAs present in the eukaryotes, whereas two different types of rRNAs present in the prokaryotes. You have to remember this numbering as well. Types of RNAs present in the prokaryotes and types of RNAs present in the eukaryotes. Large subunit as well as small subunit. And in case of large subunit, there are around 34 proteins, and in case of eukaryotes, there are 49 proteins. Okay, more proteins than the prokaryotes. So smaller subunit is made up of 16S rRNA in case of prokaryotes, and it is made up of 18S rRNA in case of eukaryotes, and it is made up of 21 proteins, and this is made up of 33 proteins. Always higher amount of proteins in eukaryotic ribosomal composition than the prokaryotic composition. But here we have very much significance for this RNA present inside the 30th subunit of prokaryotes as well as eukaryotes. So 16S rRNA, would have heard about 16S rRNA sequencing, right? So we generally do rRNA sequencing if you want to find out the unknown species or unknown microorganism. So let's say you have purified an organism from a soil. You don't know what type of organism that you have purified. You want to find out whether it is a novel one or already existing one, or it is closely related to any existing species or genus. If you want to find out, what you do is you'll go for 16S rRNA sequencing for the bacterial species, right? So this RNA is very highly conserved. RNA present inside the ribosomes are very highly conserved, because even if minor changes in the RNA structure will hamper the protein synthesis, which is very important inside the cell. That's why RNA will be never modified. It will be highly conserved inside the ribosomes. So that's why rRNA sequencing can give very good results, right? So what you do is, after doing rRNA sequencing, you will get the sequence of RNA and you will compare with the already existing sequences of known genuses and species of bacteria. Now you can find out whether it is closely related to any genus or species or any other organism, or it is altogether a novel one. Based on that, we can predict. Similarly, 18S rRNA sequencing for other eukaryotic organisms. So these are some of the major differences between the prokaryotes and the eukaryotic ribosomes. So let's move on to the next major organelle, that is cell membrane. So the outer layer or outer covering of animal cell is with the cell membrane. Outer, it is also called as cell envelope or cell covering, which is present in the animal cells. Whereas in case of plant cells, you have another cell wall on top of the membrane. But whereas in case of animal cells, you don't have a cell wall, directly you have a cell membrane on top, right? So let's assume that this is the cell interior and this is the cell exterior. Okay, this is the cytoplasm side and this is the ECM side. ECM stands for extracellular matrix side. So this cell membrane is made up of two layers of lipids and integral membrane proteins. Any cell membrane is made up of two layers of lipids and integral membrane or transmembrane protein, which is extending from one end of the membrane to the other end of the membrane. They form channels or tunnels for transport of the molecules from one side of the membrane to the other side of the membrane. So what are the other components that are there in the cell membrane? You can see a small molecules called cholesterol, which are the characteristic features of mammalian cells or animal cells. Cholesterol helps a key role in regulation of the movement of the molecules through the cell membrane. And you have the glycoproteins, you have glycolipids. These glycoproteins or glycolipids helps in recognition by the immune cells or immune molecules, which plays a crucial role in the defense mechanism. And you have the ECM components. You have the collagen, you have the fibronectin, you have the laminin present in the extracellular matrix. These molecules will be adhered by the cells with the help of integrins. So integrins are integral membrane proteins, which helps in attachment of the cell membrane or the cell to the extracellular matrix, or the attachment of cell to another cell. Cell to cell contact is also happening because of specialized structures called integrins present on the top of the cell membrane. So these are the major components of a cell membrane. So we would have heard about the protein floating model, right? So proteins floating in the sea of the lipids, isn't it? So this is how the structure look like. So here, what are the major functions of cell membrane? So first major function is giving protection to the cell as having a cell envelope, and second is selective permeability. That means it will allow only specific types of biomolecules to pass through the cell membrane and prevent all the other types of molecules to pass through the channels or through the membrane. Third one is transport of the molecules. Transport of the molecules is also possible through the membrane. So we have seen in the earlier classes, when the Golgi complex forms vesicles in the trans end, the vesicles loaded with the proteins or enzymes have to move to the cell membrane. They will fuse with the cell membrane and form a uniform structure, and release all the cellular vesicular contents out of the cell through the cell membrane by exocytosis, a phenomena which secrete or excrete contents out of the cell is called exocytosis. So that's transport of the molecules. Cell signaling. So cell signaling is possible only because of the presence of receptors on the cell membrane. So there are so many receptors present on the cell membrane. Cell signaling happens only because of the receptors present on the cell membrane. Ligand molecules come and bind on to the receptor, and the receptor is bound by a specific ligand. A particular signaling pathway will be activated. Let's take the example of epidermal growth factor, EGF. EGF is a ligand. When the EGF comes and bind to EGFR, epidermal growth factor receptor present on the cell membrane, what it do is it will signal the cell to undergo cell division. Only in the presence of an external signaling, cell division can happen. Similarly, so many phenomena that happens inside the cell is done with the help of signaling molecules. So signaling is again helped by receptors present on the cell membrane. Next major function is cell adhesion. As I said before, cell to cell contact or cell to cell adhesion is possible because of a specialized structures called integrins present on the cell membrane, and cell recognition and immune response also possible because of the glycoproteins and glycolipids present on the surface of the cell membrane. And another major function of cell membrane is going to provide a rigid shape and structural support to the entire cell. So these are some of the key functions that are carried out by the cell membrane. So I'll wind up the session here. Thank you.